So I'm very pleased to invite you back to Grand Rounds this week. My name is Rebecca Muley, and I'm one of the Grand Rounds co-directors, along with Dr. Zadie Talati and Alexander Harris. Dr. Harris is going to be our remote Zoom monitor. Before we begin today's rounds, an update on our lovely refreshments, coffee, pastries, and then our trainee lunch funding. So a huge thank you to our area leaders who have really stepped up in fabulous fashion and who have funded for the next several weeks our opportunity to share a bit of community around the coffee pot as well as for our trainee lunches. And so we're very, very pleased. And particularly for this round, Dr. Jonathan Javich as well as Dr. Jeremy Veenstra Vanderweel were utilizing their funds to provide our food and beverage. These are small. Yes, I think that deserves a round of applause. Thank you so much. Next week's speaker is our Women in Science Empowerment Lecture, or WISE. And if I can advance the slides, one. Oh, there we go. All right. So we are very pleased to say that Dr. Tamar Gur is going to be giving her lecture next week. It will be a remote lecture, but we are still meeting in the auditorium and we are live streaming her talk. So we will still have the opportunity to have community in the auditorium. So we do hope that you'll be able to join us in person. She is an associate professor of psychiatry and behavioral health at Ohio State University. We're very excited to host her lecture. Please check our Grand Rounds calendar on the Columbia Psychiatry website and watch for our Grand Rounds emails with updates for the upcoming Grand Rounds. And as you can see, after Tamar Gur, we have Dr. Mary McKay, and then following that, our own Jean Marie Alves Bradford, who will be presenting on upstander training, which I think we all need these days. And now it is my pleasure to introduce Dr. Stephan Sanders, professor of pediatric neurogenetics at the University of Oxford and an affiliate at the New York Genome Center. Dr. Sanders is a professor of pediatric neurogenetics at the University of Oxford. He also holds faculty appointments at the University of California, San Francisco, UCSF. And his group specializes in the genetics of neurodevelopmental disorders, including genomics, functional genomics, and therapeutics. His work has identified many genes that contribute to autism spectrum disorder and neurodevelopmental disorders through the identification of rare and de novo genetic variants. He has used these findings to elucidate insights into the cell types and biological processes that underlie these disorders, to investigate genotype-phenotype relationships, including sex bias, and to develop screens for genetic therapies. He directs the MRC Center of Research Excellence, or CORE, in therapeutics genomics, that aims to accelerate progress in developing genetic therapies for rare genetic disorders. He has been funded by the NIH and the NIMH, as well as the Simons Foundation. And I have to say that I've known Stephan since I started at Yale, and he is a key reason why I'm studying the gene CHD8. Maybe I have to blame him for that a bit as well. But he, along with Dr. Matthew State, really were one of the key players, along with Bernie Devil and many others. But I have to say that Stephan is one of the reasons that we know so much about autism biology these days. So it is my great pleasure to invite Stephan up to the podium.
Thank you. It's a great pleasure and honor to be here. I saw a news article that Columbia is one of the universities that parents most want their children to go to. And so I ran this past my parents, and they were very excited I was coming to talk here. In the midst of turbulent times, I'm going to give a talk today which is hopeful in the field of neurodevelopmental disorders. And we're going to talk about this in the context of therapeutic genomics and what that is. I was asked to move this. There we go. Let's start off with some quick admin. These are my disclosures. I receive funding from numerous places. I work at 3 institutions and also receive funding from BioMarin Pharmaceuticals. I've been asked to create some learning points. Today's talk will be brought to you by the letter A, the gene SCN2A, and the concept of therapeutic genomics. Let's start off with why to study genetics in these disorders. Like so many things, it begins with epidemiology. If we think about the heritability of neuropsychiatric disorders, we find they are amongst the most heritable of all complex traits in humans. This is one example of this, a nice study because it looks at multiple different traits in parallel using insurance records. This is done at the Vanderbilt system. By doing a consistent analysis across siblings, they can rank all complex disorders by their heritability, which is code for how genetic they are. And top of the pile, we find autism spectrum disorder. All of the ones in red are neuropsychiatric disorders. There is something fundamentally sensitive to genetic perturbation about the brain in neuropsychiatric disorders. It therefore follows, if we follow the trail of breadcrumbs of the genetics, we can learn something about it. My own work really began in New York with the Simons Simplex collection, funded by Safari, which collected families in which there were two unaffected parents, an unaffected sibling as a control, and an autism case. This was many years ago in Yale, as Rebecca was alluding to, and the experiment was simple. We extracted the DNA, sequenced the coding region, ran that through to get the genetic variants, and because we had the parents, we were able to identify germline de novo mutations. In our genomes, we have about 3 million variants. Of those, about 70 in each individual is a de novo mutation. They have the potential to transmit very large amounts of risk because they've not been through the selective process beyond survivability. The critical finding was a bar plot showing that there were more of these mutations in cases than controls. We've ruled out the possibility this is because of hypermutability, which leaves ascertainment. Therefore, a fraction of these de novo variants have a causal relationship to autism. The game then becomes which of those variants are the ones causing the risk and which ones are the neutral background. This is the actual bar plot of our first analysis. This was just on 200 families, and you can see the red plots standing proud of the blue ones in siblings. Red is cases, blue is sibling controls, for disruptive variants, particularly stop gain. We only had about 15 stop gains in the entire analysis. This is a table of them. And the thing which stood out was 2 of them were in the same gene, the gene SCN2A. This gene had already been associated with early onset seizures. And the strange thing was that our cases had autism, they had intellectual disability, but they didn't have seizures. This caused us a bit of hand-wringing, but with time, it emerged that we weren't wrong, but actually there was something more complex going on here. The work I'm going to show here really is at the tail end of this process. This has been a 15-year journey, and key to that has been the family group. We frankly would not have made progress without them. And here's one of the pictures of that group together. I'm going to highlight one particular researcher within that, Dr. Kevin Bender at UCSF, who's a neuroscientist who specializes in electrophysiology. And this story really represents our co-work together in trying to understand what's going on in this gene, assisted by many others in the community. So SCN2A encodes a voltage-gated sodium channel called NAV 1.2. This is its structure. You can see in the pink 4 amino acids, and in the center of that is a little hole marked with a star. And it's through that hole that sodium flows, and it's selective, so only sodium can pass through. In its native state, it is closed, and it can change conformation to open. And that change in conformation is really accomplished by the voltage sensors, which are shown here in blue. Now, SCN2A plays an absolutely critical role in our brains. In fact, it's so critical that I hope that this audience will be using this channel as they listen to this talk, and we'll get on for why that is a hope rather than a guarantee later. We find this channel throughout the excitatory neurons, but it's especially dense in the axon initial segment. It sits there in a closed state. When other neurons release their neurotransmitters and tickle the neuron, it responds by small changes in its membrane potential. Eventually, that hits a threshold where those voltage sensors react, undergoing a conformational change, SCN2A or NAV1.2 opens, allowing sodium to rush into the neuron, creating the action potential. Milliseconds later, the fast inactivation gate, shown here in blue, swings round and stops that flow of sodium. And then, of course, potassium goes out of the cell, creating the downbeat of the action potential. That action potential roars down the axon to go and stimulate other neurons. But critically, and this is the bit which was new to me when I first learned about this, it goes backwards too. So the action potential is not unidirectional, it also goes back towards the dendrites and synapses, which help to spark the change in conformation in the first place. And after all of that, as the NAV1.2 resets to its default position, ready to go again. And of course, this is happening multiple times in short intervals in our brain all the time. So that is the story of SCN2A in the early developing brain. And that's shown here on the left, where SCN2A is the predominant sodium channel for creating action potentials. However, around the age of 1 year in humans, about 1 week in mice, another sodium channel called SCN8A comes along, and it takes over the role of creating action potentials. SCN8A is a much more dynamic channel. It requires less change in membrane potential to open, and therefore becomes the predominant cause of the action potential. However, it's somewhat localized to the axon initial segment. In contrast, SCN2A is distributed throughout the entire neuron, including the dendrites. And so in later development, SCN2A's role is in back propagation of that message going back to the synapses which created it. So having understood that, and then by looking at the electrophysiological changes of the various mutations and variants we identified, over the last 15 years, we've been able to bring about an understanding of the mechanism by which you get different phenotypes. And it's a bit complicated, but we're going to go through it slowly. So this image represents the universe of potential changes in SCN2A function. We have on the y-axis the change in normal levels. So 100% is the typical value. If you're missing a copy, it goes down to 50%. If you overexpressed it, you would in theory go up on that axis. And we have seen a couple of individuals with duplications. The x-axis is the gain of function axis. And so this is a new function on top of the normal one, and that's why I distinguish it. We often think of gain of function as being the opposite of loss of function, but in this channel, that simplistic way of thinking of it doesn't work. And that's partly because gain of function is not one concept. It's many, many different ways by which a channel can start working in different ways. As we go through this, I'm going to raise the question, is SCN2A unique in this? Or is this what all genes look like, or all proteins look like once you start studying them enough? That you have a relatively single concept of loss of function, but a very large universe of gain of function mechanisms. And as we're going to see, those two can coexist, and that's why you need the two axes. So fitting that to phenotype, we need to think about the relative excitability of SCN8A. Remember, at 1 year of age, this comes along in a human brain and takes over making action potentials. And so SCN8A is more excitable than SCN2A. And so this is the theoretical position SCN8A would exist on the 2A axis. So the first diagnosis that was made was benign infantile seizures. So these are children who are born and in the first few days of life have seizures. It tends to run in families, such that the doctors are normally more freaked out than the families. And amazingly, at the age of about 1 year of life, they resolve. And this is caused by a mild gain of function, such that the increase in function is less than that of SCN8A, we presume. We're going to ignore that because these resolve without long-term neurological problems, and so effectively are self-resolving. We then have the other group of early onset seizures, and we're defining here infantile seizures as seizure onset in the first 3 months of life. And we're hoping to make this into an official nomenclature of what the DEE, developmental epileptic encephalopathy version of SCN2A is. And so here we have, at this extreme end of gain of function, a very, very severe neurodevelopmental phenotype of early onset epileptic encephalopathy. These children are severely affected, often, almost always nonverbal, often wheelchair bound, and it comes with significant mortality. Between that and the benign infantile seizures, there's an emerging phenotype of episodic ataxia. We presume that the developmental epileptic encephalopathic individuals also have ataxia, but we just can't observe it because of their challenges in motor control. In the middle, we have a group of mixed gain and loss of function. So how can that happen? Let me give an example. If you had a mutation which made it harder for SCN2A to open, or NAV1.2 to open, that would be a loss of function mutation. But if that same mutation made it so that when it opens, it stays open, that would be a gain of function. And so that one variant, if it can cause both of those things to happen, is both a loss of function variant and a gain of function variant. And herein lies the risk of thinking of gain of function as simply being the opposite of loss of function. This group of individuals tends to have seizure onset between 3 and 24 months, often around 9 months. At the severe end is often accompanied by a movement disorder. It is thankfully rare, but incredibly hard to treat because you're not trying to make the channel go up or down. You have to somehow try and find a middle ground between the both. And then the most common group is the loss of function. And this is the mutations which we found in looking at the group from the Simon Simplex collection. And so here we have, for example, a stop gain mutation, which means that one of the two copies is not working, and there's a relative decrease in the amount of SCN2A. And so this causes autism and developmental delay. And about 25% of those individuals have late onset seizures, usually after the age of 3 years. Because loss of function is easier to achieve than gain of function, we see a lot more of these individuals. In fact, we estimate it's about 6 or 7 times more common than the early onset seizure phenotypes. Of course, when we think of the family group, it's roughly equal between gain of function and loss of function, simply because epilepsy is much more likely to lead to a genetic test. This question of mechanism becomes critical once we start thinking about therapeutics. If we have a severe gain of function, then presumably we want to decrease the amount of SCN2A. If it's a mixed gain and loss of function, again, we want to decrease it, but it probably needs to be specific to the allele with the mutation on. Otherwise, you're going to cause an extreme loss of function phenotype. And if it's loss of function, we want to upregulate it. So this understanding of mechanism is the first question we need to address in thinking about a therapeutic strategy going forward. And so we are just seeing an emerging group of therapeutics which are capable of doing some of these. A company called Praxis has a GAPMA ASO, and we're going to get onto ASOs in a second, which can downregulate both copies of SCN2A. NLRM has generated an allele-specific ASO specific to one mutation within the SCN2A phenotype, which is a mixed gain and loss of function. And then Regal Therapeutics is working on a CRISPR-A approach, which again, we're going to come to in a second. And so in the space of 15 years, the community working on SCN2A has been able to go from identifying this gene all the way to the beginning of in-human clinical trials. So let's dig into ASOs for a bit, because I think these are a critical tool in our armory for helping in developmental delay. So we're going to start with some basic biology here. So here's some DNA. I spent a long time making this almost completely accurate in its molecular structure. some RNA to go with it, which is single-stranded. An ASO is simply like RNA, but the antisense. It's about 15 to 30 base pairs long, usually around 20 base pairs. For those who've done PCR, it's much like a primer. And so here's an example of the ASO binding to the RNA. Now, the thing which makes an ASO special, if I was to just put in a small amount of antisense RNA into a nucleus, it would get degraded almost instantaneously. The thing which makes an antisense oligonucleotide different is that the backbone of the RNA has been chemically modified. And that chemical modification does two things. Firstly, it makes it almost indestructible. So you can dose these by giving a single dose into the spine, into the CSF, as an intrathecal route of delivery. And that dose usually lasts about 3 months. The second thing it does is to make it clamp to the RNA really tightly. And it's that clamping which allows it to interfere with RNA. And so here's an example of the things you can accomplish using an ASO by interfering with how RNA works. And so we have an example here in the DNA space of a gene being encoded from left, and you can see the green start site, to right with the red stop codon. In the middle, we have POL2, it's shown in purple, which of course is the enzyme going along transcribing the DNA into the RNA. And in real time, we have the spliceosome, shown in blue, cutting out the introns, leading to mature mRNA, which then leaves the nucleus. The ASO can interfere with 3 things along this journey. Firstly, it can interfere with splicing by causing an exon to be skipped. This is shown on the left here, and this would be beneficial in Duchenne muscular dystrophy. To accomplish this skipping, if we think of it in a simplistic way, if you can hide the splice site, then the splicing machinery will simply skip that exon. The next thing we can do is the opposite. We can lead to exon retention. A little bit more difficult to achieve, but by interfering with the splicing machinery again, possibly by making it go a bit slower, you can encourage that exon to be included. And this has led to the therapy nusinersen in spinal muscular atrophy, which has had a dramatic effect. So spinal muscular atrophy is a disorder where children develop an irreversible paralysis, including of their diaphragm due to motor neuron death, and is almost universally fatal by the age of 2 years. By this ASO, and there are some other therapies, including a small molecule and an AAV gene replacement, can essentially create this fatal position, turn this fatal condition into a survivable one. And so we've seen children living to 14 plus years, and we expect longer. It's just that's how long these therapies have been around. The last thing we can do is by degrading the RNA. And here, there's a little black, it's going to make everything yellow again. There's a little black bit in the middle of this ASO, that's the gapmer. The middle bit is not modified, and so that triggers RNase H to come along and degrade the RNA. We ran a little thought experiment about what this actually looks like in the nucleus. So it takes minutes for RNA to be transcribed from the DNA. So in molecular terms, that is glacial. We reckon that there's probably about 50 to 100,000 copies of the ASO in each nucleus in the brain. And that means that it is interfering with the RNA almost immediately after the RNA has been transcribed. In fact, so much so that the mechanism in Angelman syndrome relies in it disrupting the RNA upstream of POL2 such that it rips POL2 off the DNA. And so therefore must be happening very, very soon after the RNA is made. All right, so ASOs are a key piece, and now we have thousands and thousands of individuals being successfully dosed with these. And so they're starting to become a bit more familiar as a therapeutic route forward. So we talked about loss of SCN2A leading to autism spectrum disorder. And so ideally, we would find an ASO which would increase the amount of SCN2A. Unfortunately, despite our best efforts, we've yet to achieve that. Instead, what we have is a mechanism called CRISPR-A, and we're going to go through what that can achieve. And here I'm going through this thinking about what is it possible to achieve in therapeutics of a neurodevelopmental disorder. This is also going to get back to why I hope that you're using SCN2A. So here we have early in development. This is in mouse. So we're before that 1 week stage where we see the SCN2A, SCN8A switch. At this stage, SCN2A is the main cause of action or main trigger of action potentials. And we see that when you are the blue mouse and you're missing one copy, it is harder to create an action potential. So effectively, you have a decrease in the number of action potentials or a reduction in excitability. But when SCN8A comes along, that goes away. So we could argue that autism developmental delay is caused by a wiring deficit induced by that reduction in excitability early in development. But if that's the case, then it seems unlikely that treating SCN2A would have an effect because it goes away. In contrast, when we think about back propagation, so this is the action potential going up to the synapses, we see that there is a deficit there in how quickly the action potential can go back up, which effectively means it runs out of steam and doesn't get all the way to the ends of the dendrites. And this is present right from the beginning of development, but actually becomes more dramatic as development comes on. If we look at the synapses, we can look at the AMPA to NMDA ratio, and this is a metric of the maturity of the synapses. We see in grey here the wild type mature synapse. In contrast, in the heterozygous SCN2A knockout, there is a change leading to an immature synapse through the lens of AMPA to NMDA. Or, put in a more simple way, it looks like this is a learning deficit. It looks like the action potential is not getting back to those synapses to train them and say, good job for firing, leading to synapses which don't mature. And this is why I hope that you're using SCN2A, because it looks like SCN2A fundamentally in an adult or a child allows you to learn. So which of these two represents neurodevelopmental delay? Is it a wiring deficit early in development, which might be very hard to treat, or is it a failure to learn at the level of the synapse, but potentially with an intact wiring of the brain, which might be treatable? So we asked this question in SCN2A mice with all the caveats that should bring, and I don't have human data to say that this is the correct interpretation. But if we believe mice, if we take this same experiment, this is a replication of the experiment we saw here, and we're going to look at the speed of that action potential going back for back propagation. That's shown again on the right. On the left is that same in a replication, we see the wild type in black and the knockout, the heterozygous knockout in blue. And we see that deficit in speed, meaning the action potential is not getting to the dendrites. Next, we gave CRISPR-A. So CRISPR-A is a way of using the CRISPR machinery to upregulate the wild type allele. If we do that in the wild type mice, we don't see an increase in speed, which might suggest we have created a problem. So it looks like you can increase the amount of SCN2A without causing a new symptom. Because we might be concerned that it would create seizures, but it doesn't seem to do that. And then in purple, this is the heterozygous knockout treated with CRISPR-A, a single dose delivered into the tail vein, delivered by an AAV virus. And you can see that it's restored the speed of that action potential in the mature neuron of a mouse. And so it looks like that failure to get the action potential back to the dendrites is rescuable. And then the second thing it does is the maturity of the synapses. So the AMPA to NMDA ratio. And the same story is true here. We see the mature synapse in black. This is early data. We have more dots on this diagram in the version which we're hopefully going to get published soon. We see the decrease in blue in the heterozygote. We see we don't get a change in the SCN2A wild type, suggesting there's not a new phenotype being created by overdosing. And we see restoration of the synaptic maturity in the heterozygous treated with CRISPR-A. And so, if the answer to what causes developmental delay in autism lies in synaptic maturity, it looks like in a mouse that phenotype is rescuable. Hope for the future. But this has taken us 15 years. And so in 15 years, in a gene, which let's be honest, is easy because we stand on the shoulders of 30 or 40 years of work into what sodium channels do. But in 15 years, we've been able to apply these genome-targeted therapies, ASOs, CRISPR activation, and start to see the first hints of achieving clinical benefit. But that's one gene. There are 4,500 single gene disorders. Oh, I've put a UK slide in here. Imagine you're in California. It affects 2.5 million people in California, or approximately 500 million globally. And this is the weird paradox of rare genetic disorders. Individually rare, but collectively common and frequently devastating. And so it's been estimated at the current rate of progress, we should have this licked in 2,000 years. And it's been estimated that will also cost approximately the GDP of the whole of the United States. So how do we possibly apply these tools? We are in an amazing position right now. For the first time in human history, we actually have a suite of tools that could make a difference. In the last 15 years, but really in the last 5 years, we've seen these applied to humans with some absolutely spectacular successes. Here's a list of genes and disorders where we've seen what looks almost like a cure delivered in some of these through gene replacement, through CRISPR editing, some of it in vivo, and through antisense oligonucleotides. And so this is just an amazing time to be working in the field of rare genetic disorders. But each of these cost millions and took years and years and years to make. So back to the question, how do we develop this for 4,500 disorders? Unlike therapies that have come before, these genetically targeted therapies have some really unique properties, which I think makes this possible. So the first one is they are very flexible. In theory, you can target almost any variant, often with some of the tools we have already. By changing the targeting sequence of the genetic medicine, you can get it to almost anywhere in the genome or anywhere in RNA. In theory, they should be able to treat most disorders, not all, but most. In theory, they should be more specific. So we think that lots of the side effects come from off-target effects of small molecules or antibodies. If you want to get really specific, then genetic hybridization, DNA base pairing, is one of the best ways of doing it. If you can get 20 base pairs of DNA, that is unique to a single place in the genome. So in theory, these should be able to target one gene and one gene alone or one location alone, and therefore hopefully bring fewer side effects. Of course, the reality is that you have to demonstrate that they are not causing lots of off-target effects, which still remains a challenge. But in theory, we get there. But the most exciting one is this last one, the reprogrammability. If I create a small molecule, and it takes 15 years to do it, and then I want to do that again, you're largely back to the drawing board. You need to start from scratch with the preclinical work to work out how you're going to make a molecule to target a different process. With a genetic medicine, if you can simply change that targeting sequence, you can inherit all of the information you had on the last successful therapy. And so in theory, you could reprogram it to the next disorder quickly. Sounds good in principle, but actually it's already happened in practice. So we're going to show two examples here. The first one, on the left, we have that ASO for spinal muscular atrophy. And that was reprogrammed by Tim Yu at Boston into a therapy for Batten's disease. All he changed was the base pair sequence. And this took 16 months from diagnosis of the child to the first dose of intrathecal ASO, including going through all of the FDA regulation. And it was because we knew so much about what ASOs do from spinal muscular atrophy that that success was able to be achieved quickly. And at the bottom, we have work by Intellia. This is in vivo CRISPR editing in the liver. And so on the left is a therapy for transthyretin amyloidosis, essentially knocking out the gene. That took a long time to develop. But on the right, 2 years later, they were able to reprogram that into a therapy for hereditary angioedema. Same tissue, but completely different gene, just by changing the targeting sequence. And so this idea of reprogramming already exists in clinical practice in humans. The question then is, can we harness this at scale? Can we imagine a world where I see a patient in clinic and then can predict the ASO or the CRISPR edit to treat that child, maybe in theory, the same day, excluding the problems of manufacturing? And it's on that basis that we coined this term therapeutic genomics. The idea of harnessing the power of the genomic approach towards data and experimentation towards therapies. And we recognize it's not just the technology, it's the philosophy that goes with it. Fundamentally, this is about sharing and about the collaboration between the informatics and the therapeutic space. We need to design every single experiment, so not only is it advancing the therapy being developed, but is also generating the knowledge base to treat the next individual. If we think about what genomics has achieved, this concept has already worked. And so let's start off with genomics. If we go back 15 years when Rebecca and I started working together, we were working on candidate gene analysis. You had to take about 200 individuals diagnosed with a disorder. You had to pick your gene with no knowledge of whether it's the right gene or not, and then PCR every single exon separately in those 200. It took 2 to 3 years if you were lucky per gene. But then with genomic technology came along, instead of doing small populations in one gene, we did large populations with all of the genes in parallel. And it wasn't just about the technology, it was about people working together. It was about the data being systematic and collected and shared. And with that, we were able to start finding things like SCN2A. So here's the mutations, the de novo mutations we see in controls, and they're scattered across genes. This is a little section of chromosome 2. In cases, though, we see these mutations stacking up. And so this is how SCN2A looked a few years ago. We found 2 initially, but rapidly that grew. And so the way to work out, well, which risk variants are the ones which are contributing is by, do they stack up in a gene? There's a whole complicated piece of statistics behind this, about how many variants, size of the gene, mutability. But it basically comes down to this. With enough mutations, you can identify the gene. And so in the space of 15 years, that's been applied to identify hundreds of genes associated with neurodevelopmental delay, most of them not recognized as causes of human disease before this era. And this is the list of the 373. These are all genome-wise significant. The size of the circle represents the number of individuals identified in a cohort of 80,000 plus individuals. And it's no surprise that SCN2A was one of the first genes found, because it ends up being one of the more common causes. CHDA is just there, in case you're and so all of these top tier ones here, these are quite common disorders, so much so that they should be suitable for some therapeutic development. Of course, this progress has not been just in the brain. In fact, it's happened across multiple different rare diseases, retinal blindness or immune disorders, such that in 15 years, we've gone from having these broad categories to having all the preclinical information, but also the clinical application, that now many individuals are given a gene and a variant as their diagnosis. And so it was through that philosophy of sharing that this was possible. We're in the middle now of the second phase of this, a functional genomic revolution. And the same paradigm is true that we used to look in individual tissues, in individual genes, for example, a qPCR experiment. And by applying single cell sequencing or spatial transcriptomics or all the other amazing tools we have now, we can look in multiple cells and multiple genes in parallel. And we're now seeing emerging efforts of integrating that across individuals, because the data is systematic, and is shared between different individuals. And this data is directly relevant to therapeutics. So here, going back to SCN2A, here's a map of some ChIP-seq data and ATAC-seq data. And from this, we can see these peaks pulling out the promoter regions. And if we look hard enough, we can find some putative enhancers controlling this. So we start to have a map of the regulatory space around this gene. We can look at splicing patterns. This is the difference in exon 5N versus 5A. We see a developmental shift in exon usage in SCN2A, with interestingly the shift happening around 1 year of age, speaking towards that same shift of SCN2A and SCN8A. This is showing this for SCN2A alone, but we can also start applying these data sets to start looking at global questions. This is a map looking at the expression of autism genes across development in bulk RNA sequencing data. It shows that there's more data, there's more expression of these genes early in development than late in development, which leads us to think this is probably a prenatal disorder. However, with single cell sequencing, you can get a more refined analysis of this. If I take the 72 genes associated with autism in a 2020 analysis, we can split them into two large functional categories. In purpley pink, we have the genes associated with transcriptional regulation, for example, CHD8. In green, we have the genes associated with neuronal communication, for example, SCN2A. If I show that same pattern of these across development, split by gene function and split by cell type, we see this. Work in progress, probably it's not as pretty as it could be. But on the left, we have the transcriptional regulators, and we have the excitatory neurons here, which is where we think the action is taking place. There's a slight bump in the prenatal space, but not very much. In contrast, if we look at the genes associated with neuronal communication, we see a very, very different pattern in the excitatory neurons and the inhibitory neurons. The highest expression is after birth, which then plateaus. And so, on the theme of hope, this is another slide that gives me hope. This makes me think more towards the idea this is a synaptic learning phenotype rather than developmental wiring phenotype. One thing which we're trying to ask is, can you show that the transcriptional regulators are also acting at the level of the synapse? I'd make the observation that there seems very little clinical difference between these two groups, making me think that actually they are both working through the same mechanism rather than different ones, which seems more likely to be at the synapse. And this is work done by a postdoc in my lab. And so this functional genomic revolution has shown us that we can understand gene regulation at scale. And that's directly relevant to therapeutics, because if we think of how many of these work, it is through understanding the gene regulation and then manipulating it that you can make a difference, either through the generation of the RNA or by modifying splicing. And so then on that background, we coined this term therapeutic genomics, and we want to try and achieve that same progress, going from developing one therapy in one gene to multiple therapies in multiple genes in parallel, by harnessing the same idea that if we can start collecting this data in a systematic way, sharing it widely across collaborators, that hopefully we can build the knowledge base to predict which guide and which ASO will work in which context. And a group of us in the UK at Oxford, but also UCL, Newcastle, UC Berkeley and UCSF, recently received some funding to try and bring this concept to life. And we're going to split ourselves into 5 groups. The first one being recognizing that this is fundamentally an informatics problem. And so we're going to try and generate the informatic tools we need and then, of course, share them widely with the community. The first bit is about trying to identify targets. And so we started to create this gene-level database called GeneDB, which we're close to being able to release into the wild, which simply integrates all of the useful data sets in this space. Behind the scenes, it has a trick to keep it up to date as the gene names keep changing. And with this, we can start to build maps, which we've always wondered about, but never really been able to do. So here's a map of the genes associated with developmental delay by mode of inheritance and then by mechanism. Loss of function, toxic allele, that's the phrase for gain of function, trying to use a more, trying to create the difference between toxic and loss of function rather than gain of function. And then the mix, which is a mixture of loss and toxic allele. So here we have that first critical information of mechanism and mode of inheritance when we start thinking about therapies. We're working on expanding what ASOs can do. We've seen them work with a poison exon, and we've seen them work with disrupting genes, but there are other methods coming online, including enhancers, upstream open reading frames, and 3' UTRs. By collecting data on how each of these work now in a systematic way, as we start to see the first successes in this, we should be able to predict which genes are the most suitable for which different methods. And on predicting therapies, this is a map of CRISPR edits in inborn errors of immunity. And so this is work done by James Davies at Oxford, but also the UC Berkeley team has some very similar software. And it's taking the pathogenic variants from ClinVar and asking, with the existing CRISPR editing tools, what fraction of them could be fixed in theory by Cas9 HDR or base editing or prime editing. And what we found was that already with the existing suite of genome editing tools, about 70% of variants have at least a first approach of a guide that could create an edit to go back to the wild type. And so this tool, which MEdit, which is the UC Berkeley one, should be released any day now, allows you to take a variant and predict the guide which will work to treat it. So that's the informatics piece. We're then going to start this platformization and reprogramming, and we're going to start in the blood and the eye with CRISPR. So one of the interesting things in this paradigm of therapeutic genomics is that what happens in the blood is now directly relevant to what happens in the brain. Because once you start thinking through the lens of a targeting genetic medicine, actually you can learn a lot of the rules from a blood-borne disorder and then just port them into a different tissue type. Of course, you need to do a little bit of learning to adapt them. So in the blood, we're going to be working on EBV handling disorders, and we're going to be trying to do ex vivo genome editing. So cells are taken out, edited, and put back in. And this has been in the approach used in sickle cell. We hope to evade the need to deplete the bone marrow. In the eye, we're going to work on retinal blindness, because this is the easiest route for doing in vivo CRISPR editing in an organ where you can see it. And so you can make an injection into the retina and then observe it by looking through the pupil. In the brain, we're not quite ready for CRISPR editing because of the challenges of delivery. And so we're going to focus on ASOs and trying to learn how to predict which genes are targetable today. And to do that, we're going to start thinking about genomic screens. So again, this idea of trying to bring the genomic technology into it right from the get-go. This is work by Jason Dure and Nadav Aitov at UCSF and Seattle. It's a high-throughput screen for CRISPR activation. So we talked about developing a CRISPR activation therapy in SCN2A. That took 2 to 3 years to accomplish. This experiment uses barcoded guides, and every single dot here represents one of those guides being tested using single-cell RNA-seq. We're looking to see if we can increase the expression of TCF4, and this guide here, this is the one which achieves that the most robustly. So what makes this exciting is that this experiment was done not just for TCF4, but for 9 genes in parallel. And this was the pilot because we're now working on the full analysis of this, where we tested 300 genes in parallel, and we're able to create the first guide for 200 of them in one experiment. So we want to adapt this idea of a genomic screen for therapy to use it for ASOs and also for CRISPR editing. And so an example of doing that is instead of using Cas9 to target the DNA, we could use Cas13 to target the RNA, and then we can start working on something which looks like an ASO, but is usable in a genomic screening paradigm. And then lastly, we're going to work on brain delivery with some new approaches towards achieving this. This is obviously a very, very hot topic. Looking at where things are, I think we are a few years away at most from having a workable brain-wide delivery vector to enable the CRISPR approaches. And then across all of this, you can't write a grant without mentioning AI. But here, I hope it's clear how that's really like the heart of this. We are generating this data to be ready for AI. You get what you put in, or garbage in, garbage out. We need to, as we design these experiments, make the data ready to be used for training. And then that goes back to the informatics piece, completing that loop. Our charge is to go beyond just making these therapies, but we need to enact research culture. And so this includes trying to create a regulatory environment where this is possible. At the moment, we have commercial opportunities for the most common ones. We have NLRM and similar working on the most severe individual ones. There is a gap in the middle where we don't have an effective regulatory model. In the UK, we have the advantage of an independent regulator. We also have NICE, which sets economic thresholds of what is good value for a therapy. And so we're working with both of these to start thinking, what would this look like? Fundamentally, regulatory is designed for one molecule at a time. It doesn't allow for a mechanism by which you have the shared data from the last successful therapy. And that's what needs to be built in the middle here to make this work. And so in that research culture piece, this core represents the middle of a large ecosystem of people contributing towards this, partly in the UK environment, but also with our Berkeley and UCSF, also working with the FDA. And our hope is that by sharing all of the tools that come with this, we can create an ecosystem where we're working with everyone, including dear friends at Columbia. And the end result of this, we hope, looks like this, that there is a website you go to, you put in your gene, you put in your variant. It tells you the mechanism, it tells you if there's an ASO possible, it tells you the guide for your CRISPR edit, whether AAV is possible, and ideally, who to contact to get that therapy. And then as we think about what's going on in the generation study here, we imagined creating a world where even that isn't needed. That it all starts with newborn screening. This leads to automated diagnosis, but diagnosis is now accompanied by the design of your genetic medicine. Most of it's been manufactured already. You just create the genome targeting piece specific to the variant identified. There's a baseline level of screening because most of it's been screened already for toxicity and therapeutic efficacy. It gets sent around the country or around the world, such that the therapy goes to where the patient is. And then when the parents go in to receive the diagnosis, in the next room is their therapy. And if we think about spinal muscular atrophy, this has already been accomplished. We just have 4,499 other genes to get to. And I'll end there by thanking the wonderful team working on this and the institutions involved. Thank you very much.
Thank you so much, Stephan. That was such a lovely demonstration, and I think really gives those